What's up? What's up? Gotta make sure everything's clean and ready to go here. Don't mind the glasses. Sorry. Nerded out. One second. Good thing I got the dogs. Keep an eye. All right. Gotta close. All right. Just wanted to make sure it was working good. Good. Hey, everybody. I really appreciate everybody who's supporting us and showing up. Grateful. It's not going to start till 10 o'clock, but I like to be ahead of time. So just want to start out by saying thank you so much. I'm so grateful for everybody who supported us, coming on, subscribing to YouTube. It's awesome. Super grateful. I feel like I'm going blind. It's fucking awesome. All right. So until people start showing up, it's going to be a little bit. Like I said, I'm definitely early, but I'm excited to get some fire into the hands of all of you guys. Times have been a little bit crazy, so everybody's kind of like holding out right now. And looking at my notes. Again, just super excited. Super excited. I want to just check this out. Yes. Hopefully you guys can hear me. Sweet, what's up? How you doing, brother? You're early even. That's even better. A standard protocol for seed starting medium. I kind of have developed one that's proven very successful. It's one that we're using in tissue culture, and come to find out, it ended up working really good for seedlings as well. So I do have a specific one that's been very good, but the whole beauty of teaching people is, do I think it's the best? No, I think someone could create something much better, but it works. Yes. What's up? Thank you for joining. I'm so grateful for everybody so far who's showing up, who's subscribed to our channel. Fucking awesome, man. This is something that I've literally dreamed of. Something we're going to talk about. It's really awesome because a lot of the stuff that I've got on my little notes I've got written here, I've thought about, and it's come true. It's crazy. Yeah, so the medium is definitely a standard protocol. I have something that works. It's a rooting medium, basically TDZ, NAA, and some indobutyric acid, and a little bit of charcoal in the medium. Works awesome. Happy to share that recipe, because it's the recipes. Everybody's kind of like always asks that, what's up with the sauce? What's up with the medium? The recipes are really, it's exciting because there's no right or wrong solution. You got to experiment with every plant, every strain. And the whole beauty of it is, it's like you'll never get to the top. You're going to keep, there's so many variations and experimentation with solutions that you can really create special stuff. You can create special protocols and solutions for each plant that make them just perform like I've never seen. It's one of the funnest things I've ever done, because it's like you can get really, really creative. And when your ability to create shows fruition and things do good, it's a fucking trip. It's one of the best things I've ever experienced. That's why I want to share what I've learned with other people, because it's fun what I'm doing, but it's even more fun when I can share with other people and help them Honestly, it's about serving other people, watching other people succeed, helping them succeed, follow their dreams. That's how you build your own. It's incredible. Anyway, sorry, I'm not paying attention. I'm already flapping my lips. Really old seeds, I find that they fossilize. This is the crazy thing about old seeds. It's not that they're dead on the inside. Some are, but for the most part, I find that the embryos are in good condition. It's the shells have fossilized over time. They become super hard, almost like to protect the embryo. And even if you do soften them, they tend to never poke out of that shell. I found that adding a little bit of gibberellic acid, some kinetin, a cytokine into the water with the peroxide in sterile water solution, that seems to make them pop. And again, the beautiful thing about this TC seed method is if I can just get a tiny little turtle head to poke out, that can be difficult to put in a regular medium. But in agar, if I've got that agar prepared, I can just poke that tip right into the medium and it's fucking takes off. I've saved so many old seeds there's no way I would have been able to salvage, in like a peat moss cube or soil or a paper towel. Sometimes I've had good luck doing that, but this method has proven successful over and over and over. I can salvage. It's too good now. I have so many freaking genetics. It's, a little bit of loss. It's not a bad thing. I have so many, I don't have any room now. So anyway, I'm rambling. Hopefully that answered your question about old seeds. We've salvaged some extremely old seeds. It's crazy. It's a cool method. What up, bro? Got a lot going on. A lot of interesting stuff. I'm not gonna go too long tonight. It's late. Just wanted to get a general, kind of a layout, some groundwork, what we're gonna do. But we're gonna be giving all kinds of information out on this channel, man. I'm looking forward to really people who are interested, genuinely interested in bettering themselves, in growing, and personally as well. I think I've got some stuff I can help. It's helped me tremendously. I'm looking forward to helping my fellow growers, no strings attached. Hopefully, if you guys ever decide you want to take class or gain some knowledge, you'll come to us for answers. I'm having a lot of fun doing it, so I'm looking forward to sharing with other people. Some of our students, man, have progressed. So that's been one of the best things is watching some of the students, how far they've come. It's awesome. We're going to be starting a mastermind group. Anybody who's taken the class or been a member of Black Sheep, it's going to be invite only. I'm kind of going to be a stickler because I'm looking for certain motivated people to create a group of individuals who we can hold accountable and have some really crazy sounding achievable goals that we can, as a group, work on. So that's kind of an exciting thing we're going to be working on as a mastermind group for tissue culture and people who want to improve not only their grow, but their lives. Anyway, don't mind me if I go off topic. Super excited to be here. We're early, guys. This is great. I'm so appreciative you're here early. Thank you. Am I stuttering? I may have to speak up to the camera. Is that better if I get closer? Hopefully I'm not breaking up. Give me some feedback here. Is that better? Can you guys hear me all right? Hopefully you can. Hopefully I've got good connection. It looks like I got green here, but I can talk louder. It might be echoing in my room a little bit. Good to go. Awesome. Hit me with questions if you guys have any. I'll go off and start rambling on shit, and I don't want to go off too far. If you guys have specific questions, hit me. Happy to answer. Everybody who's on who didn't get to hear it, first of all, super grateful for anybody who's jumped on and followed us, subscribed. I cannot wait to get you some fire genetics out. There's nothing better watching our creations go to people who appreciate them and make them do wonderful things. So I've got some really awesome crosses I'm going to get out to you guys. I'm happy to do that. Once I have everybody's information, I'm going to go tomorrow and put everything together. It should be Wednesday. I'll have everybody going out. So I'll get some FEMs, some regs. I got a bunch of rainbow belt crosses, a bunch of wedding pie crosses, banana OG crosses, some old school TK91s. I'm going to throw you guys all kinds of fire. Got to promise though one thing. I want some cuts back, or if you guys find something really special, that's the beauty of what we're doing here sharing is I want something back. Hopefully if you guys find some fire, I know you will. Trying to stay on this. All right. So you guys, there were some questions about really old seeds. We've been able to salvage definitely some older seeds. In the medium that we're using is a rooting media, a specific hormone blend that we're using. Getting them to soak in that water now. I literally let them sit in the hydrogen peroxide in the water solution until they just pop out of the shell. I find that if the shell is still on, I can put it into the medium, but it can't touch the medium. If it touches the medium, the shell itself, it tends to get infected. The embryo itself, never a problem. But the shells, when they fall off inside of the culture, if I do seeds in the container, they tend to fall off. And when they hit the medium, they grow fungus. And it just tells you there's no doubt that the seeds themselves are very porous and they're full of all kinds of bacteria and shit. It's evident because no matter how long I soak them or sterilize them, the embryos are good, but the seeds will grow some funky shit. They'll grow like a cap of just nastiness. So they're porous, they're full of all kinds of stuff. Soaking them in the water peroxide solution and getting them to pop out of the shell completely, awesome. The embryos aren't they don't have any infection. That shell does. So, but getting old shells to pop can be difficult, no doubt. Rainbow African haze. I don't know if it was African haze. I think it was something else. I don't think it was very good. I don't remember what we did with it. I've had so many strains. I apologize if I literally. The beautiful thing about TC is I get to keep all my strains. The shitty thing is it's like a woman in shoes. I got all these fucking strains and I'm like it's a massively overcrowding. So it's a pleasure to give out some genetics and some beans to you guys because I'm 52. There's no way I can go through all this stuff, and I'd rather have it in good hands of people who appreciate some good fire. That's exciting. So, all right. Yeah. So the Rainbow African, I think I grew it out in the summer of last year, and I'm so picky. I've gone through so many strains now that, it really has to be something special now. Otherwise, it's just not worth keeping. And I'm not really about yield. And I've become more about yield now because a lot of the people we're working with, obviously, to get a yield, that's your success. But I care about the terps. I've done this in the beginning because I love good weed. And this has really created an ability for me to just really focus in and dial in and grow some of the awesomest shit. And then the breeding factor on top of that, because I can keep my males and I can keep all my stuff forever, basically. And there's no contamination, no vectoring because they're in test tubes. No matter what happens, if I have something go wrong, I can throw it out, go back to my culture lab, bring out fresh, healthy cuts and start brand new every time and never have problems. It's one of the best things ever because I can eliminate all the problems before they begin. Fucking awesome. All right. Hopefully you guys can hear me. All right. I would love a cutback. That's the beauty of sharing. Hopefully I got some peanut butter ice cream cakes we're going through right now. Damn, there's wedding pie and banana OG, wedding pie, rainbow bells. Like I said, I have so many. It's literally hard to keep track of now. But again, everybody who's joining on early, thank you so much for participating. I'm super grateful. It's early. I just wanted to get on here. I'm so excited about, sharing some information, some genetics. I've been doing this since I was 14 years old. I hope you guys don't mind. I'm going to tell you a little bit of my history, how I got started. I was moving packs like in the 7th grade. It was crazy. $600 brown seed packs, this shit, but long story short, I was literally rolling up 70 dubs every day before first period, like 70 joints I was selling every day before class. This went on for quite a long time. And I was doing 3 for $5, just raking it in, making $300-400, a week, a month. It was just phenomenal. My family was tripping. They were so conservative. They were really tripping on what I was doing because they saw I was a businessman. So in one hand, they thought it was cool. On the other hand, they're like, this little shit. Anyway, they ended up buying me a Phototron. If you know what a Phototron is, it was a little device they sold in High Times magazine back in the 80s. And it was a little indoor grow. You could grow, weed, year round in your house, and it was like a little contained unit. It's crazy. The thing that caught me was after you grew in this thing for like 60 days, you could send a soil sample in to these guys, and they would tell you exactly what your plant was utilizing and send you back a custom-made nutrient solution so you could, have like this really particular dialed-in sauce solution. I thought that was the best thing ever. There's no doubt this guy was not testing this shit. He was sending back some veg bloom. I guarantee you. They weren't testing shit back then, but the whole thought of it was inspiring to me. And I love the fact that I learned to grow in something so small, a very small, contained, very closed area. And look what it turned out. I'm growing plants in test tubes now. But it was really cool to be able to come home from high school and pull some nugs off and microwave them. Yeah, it was cheesy, but compared to the brown brickweed back then, it was still better. Ask Micah Disney, my good friend. He'll tell you that he got so high, he turned all pale on me, and it was nothing crazy. He got all pale, got all paranoid, and quit smoking weed because it was so potent compared to the regular shit bug we were smoking. So, true story. Mom bought me a Phototron. I was like 14 years old in high school, growing weed. That was the beginning. Once I did that, it was like that gave me the taste of terps. I wanted to grow. I wanted to educate myself. They actually bought me a bunch of books on horticulture, and this one they bought me was How to Supercharge Your Garden. It was awesome, man. I went through that hydroponic stage for a while. Nothing against hydro, but supercharging with high pressure sprayers and water cooled lights, it was just nuts. Now I like simplicity. I just like good weed. Even the big bros are a little bit crazy to me, but yeah, I get it. Everybody's trying to do their thing. I just like the craft. I'm really about the craft. I think it's going to go back to that where people appreciate just really good marijuana, and what we do really allows you to do some crazy shit to these plants. And it's not genetic modification yet. This is just chemistry. Understanding chemistry inside the plants, inside of culture is one thing, but you can also create solutions now after and put them into your food and make these plants just do some nutty shit. I mean, really, you can get some yield potential. And it's not no weird chemistry. It's actually just getting the plants to grow more structure in a faster period of time, and they can support more weight. Plain and simple. Engineering. We can engineer the plants with chemistry and make them support more weight. And it's even better when they have no endophytes, no bacteria, no hops, no fusarium, no botrytis. When you have plants that don't have all that shit, you'd be surprised how fun it is to garden. It's actually fun to grow when you're not fighting every element known to man. It actually is enjoyable to garden again. Anyway, so I hope I'm rambling off here. Everybody who showed up early, we're early. You guys are awesome. Everybody who shows up early, give yourself a high five, because normally in this business, you know the deal. Day late, dollar short. So you give yourself a fucking high five, honestly, that you showed up early and we're going. I'm gonna ramble for a bit here, like I said, and challenges. That's one of the notes I've had here. How challenges. Everybody's a challenge right now. I guarantee you, man. I talk to so many people. The beautiful thing about what I do is I get to talk to a lot of people that grow in the industry, the black market to crazy large facilities. Everybody's going through stuff. Don't feel like you're alone. I can tell you that the majority of the industry, I make memes about it, of course, but sometimes you got to laugh. But the industry, everybody's feeling this, man. Everybody's got beans. Everybody's got fire. Everybody's got raws. It's kind of crazy. So the real question is how are you going to separate yourself from everybody else? What are you going to do to make yourself stand out? How can you improve yourself, your business in tough times? Sometimes when it's tough, the tough get going. And don't mind my cheesy expressions, but I'm telling you, check out the book, 6 Habits of Growth. I posted it up the other day. It's freaking awesome, man. Life-changing, literally in a short period of time. I can just tell you in a short recap, 4 things that this guy has helped me. I've already been working out for quite some time, but literally working out in the morning, followed up by 15 minutes of reading, actually physical reading. After the reading, 15 minutes of goals, even if they're, I write them every day. So I rewrite them, go over them, study them for 15 minutes. And then after that, it's meditation. And I think about it and I try to see myself in the future of all these goals that I've tried to achieve. And it's been crazy because it's really, here I am. This is one of them, being in front of you. So tough times create tough men. Tissue culture has opened up so many other opportunities for me. It can do the same for you. You know, everybody wants to have 200 strains, but I tell you, trying to keep 200 strains, it's a full-time job, man. And I'll tell you this, the biggest thing about growing is you become a slave to the garden, to your plants. You can't leave. How many relationships I've messed up because, man, I had to go home and water the plants, or the people I let water them fucked them up, and we lost that friendship. So tissue culture has given me the ability to be free, because now I can leave these plants in here, and once they're in culture and past the infection stage, it's crazy. I can literally leave for 2 or 3 months if I wanted to. They won't die. There's no watering, no spraying. It's once I put them in tubes, don't get me wrong, there's maintenance. I can have enough work to keep me busy 24/7. It just depends on my outcome. But if I don't want to, I can literally just store them, especially if it's in a colder situation. I can literally store these plants for such long periods of times. It's incredible. It gives me freedom. I can leave, come back. My strains are still there. It takes about 3 to 4 weeks to get them out of the test tube, hardened off, and to regrow into a mom again. At that point, when I'm growing moms now, I'll only usually clone off a mom twice, maybe 3 times. And at that point, from my experience and my knowledge, it's better to toss the mom. Once you start topping and cutting a plant so much, it literally alters the hormones, and all the clones you take off after that, they're whack. That's what happens after you take and take and take. They can do it. Plants can survive for years and years, but the whole point is if you just keep the mom for a limited time, you can literally experience 30 to 60% increases in performance, yield, and terps, because the plants don't get that drift. So TC is good for that. I love TC for viral elimination. That's a phenomenal thing. But really, it's about putting all my strains into culture now before they have the opportunity to get sick. That's kind of where I'm at. It's keeping the strains from getting the viruses and getting sick now. So once I start seeds, once they're about the 8th week, I'll usually start taking pieces off of the plants and putting them into culture before they're even tested. That allows me to run the plants. If they're keepers, awesome. It's already in culture. It's protected. If not, toss it, move along. Let me answer some questions here. I'll keep rambling on. All right. I know there is. Every time I look back, there's so many strains that things have changed so much now too. And I'm not hating on the strains that are around today, but I feel like everything's so cookie cutter. This is why I'm excited to teach people tissue culture, because I think when people understand the amount of knowledge available, that you can really alter strains, and it's like you really want to work with clean strains to begin with if you're breeding. I have a feeling that a lot of people have bred stuff that they didn't know. Not intentionally. I don't think anybody would do it intentionally. If you do, then you're a fuckhead. Excuse my French, but if you know you got sick plants and you're breeding, that's pretty shitty. If do the right thing, you have all your stuff tested, you know it's clean, and then you breed, you're going to end up with a much better breeding stock in the long run if you're paying attention. So I think if, for the long run, tissue culture is the answer, is because it allows you to do whole plant testing. I can take that whole plant, and if you're not testing the roots, you're not getting accuracy. Look up Toomey Genetics out of Colorado. They're one of the most accurate companies now for hops, fusarium, botrytis. Whoever's doing your testing, I swear to you, it's so important that you know who's testing, because if they're not testing your roots, they're just taking your money. The roots are where the problem lies, from my understanding now. The root mass is where the hops, especially the fusarium, botrytis, the leaf stems, the petioles are variable. Those are kind of like random, but the root mass, that's where the problems lie. So people really need to pay attention to the testing. If they're not asking you for roots, find somewhere else. Toomey, giving you a little shout out. I think they're like $25 a hops test, and if you want to do all 3, it's $60 for a full panel, which is well worth it. $60, and then you're guaranteed. And what we do now when we give out genetics, I'll usually give people 2 test tubes. And what you can do with that, you can send one off to Toomey, have it tested, get your pathology, and then you have the other one waiting at your grow in the other test tube. Once you get your pathology back, the plants are from the same tissue culture piece. So if one has it, they both do. So once you get your pathology back, it's literally insurance. It's a guarantee. What else can you get a guarantee on nowadays? This is something you can do. It's literally for $25 to $60. People can prevent all these problems that were killing us in the past. So the beautiful thing about TC is it can cut your cost, it can increase your terps, your performance, and stop loss. And everybody thinks it's crazy expensive to get involved in TC, and I assure you it's not. The equipment is relatively inexpensive in comparison to everything else in cannabis. So, and we're happy to help you with that. We don't try to upcharge prices or help people, or hack people on equipment. We just try to lead you in the right direction. My job is to educate. I want to teach you how to understand hormones in the sterilization process, how to cut a meristem properly, what to look for. The biggest thing in our education program, honestly, is the follow-up. I wish I had some students here that you could talk to, because in my experience talking to them, they've said, the class is great, but it's the follow-up afterwards. And when they have all the questions in their own lab, when they're experiencing all these problems and things come up, this is why I became a teacher. I did not intend to become a teacher. It happened by accident. I became, so frustrated after I took class that I was, pulling my hair out for almost 2 years. Felt like a dummy. Sucked. So during that process, I really, dug in, but I had people online, other people helping me. And long story short, I ended up figuring it out. But once I did, I was like, that's the ticket, man. It's literally helping people after they take class. Class isn't the big deal. We're there, we're helping you, everything's hunky-dory. It's when you leave and you're having problems and you're having issues with your media or things are coming up and you don't know what to do. When you have someone to talk to, it helps. That's how you get over the bumps and it gets you along faster. All our classes is faster. Anybody can learn this. Everything's available online on YouTube. You can find all this information. Shit, if you study my videos long enough, you can see the whole process. Really, it comes down to understanding the medium, understanding the protocol, the process. After you cut enough meristems, that's not a big deal. Honestly, the meristems are, it's more the medium, getting the correct medium to allow the plants to grow rather than making the good cut. But the better you make the cut, the better success rate you get. So anyway, again, rambling on here. Don't mind me. Ask some questions, you guys. I'll be happy to answer. I get to flapping. Greetings, Slow Speed. What's up, brother? Sorry, I started a little bit early, but I'm going to go on. The show goes on no matter what. I just wanted to get on here and get started a little bit early because I was so excited to share. I have so much information and so much stuff that I want to get out, and it's awesome that I have this platform that I can share with you guys. I can share information. I can share genetics. We're excited for the new year. Honestly, as crazy as the world is right now, we're actually excited because the possibilities in TC, some of the people we're working with, this mastermind group we're going to be starting. We've already got 3 people. We started on Thursday. If it's something you're interested in, you shoot me a DM or email. We're considering a lot of people. I'm going to cut it off about 12 people to start. It's going to take certain people with motivation and attitude and desire, no negativity, no weird shit. So people that want to improve their life, they want to improve their grow. Sometimes it's nice to have a group of people that hold you accountable. Once a month, you're on the hot seat, meaning if you have problems or issues, we all work on it as a group. And it's really crazy the amount of energy when you have goals other people actually care about, hold you accountable to. We hold ourselves accountable to. It's powerful. It's one of the most powerful things I've ever been a part of. I did this in real estate and it was life-changing. But now I love cannabis, so I want to share it with all you guys. Everybody who's on here right now, I want to share some shit with you guys. Hopefully it'll change your life. It'll inspire you to do something. Like I said, growing's fun. This part, the scientific part of TC, man, if they would have had shit in high school, I'd have been a straight A student. My phototron would have had like double the yield. Would have been crazy. So yeah. So anyway, I'm going to get back to thank you for everybody who's on here. I was super grateful for everybody who's jumped on early. Again, give yourself a pat on the back if you showed up early, because in this industry, it's usually late, day late, dollar short. So awesome job. Like anybody who's here early, you guys are on it, man. That means you're looking, you're maybe hoping, trying to achieve, give yourself a little bit of an edge. Good for you. I was just saying that challenges make us stronger, and literally we're all dealing with challenges, everything going into the holidays. This is why I'm really excited to give out some gear. Hopefully it helps some people out. They can get some fire right now. That to me is the market. You got to have some good shit. It better be good. Better be fire. so hard now to have just the average Joe because everybody's got good shit, or a lot of people I get to work with, fortunately, they have some good shit, and it's like, damn, makes me really want to step up my game because these people are bringing their A games now. And by the way, congratulations to Trichome Tortoise. Looks like he took home some gear, crushing it. Always appreciate people who got ambition out there, hustling, trying, even when things are tough. It's awesome. What's up, Jose? Thanks for joining us, brother. Everybody, I'll try to keep track on the chat here. Don't mind me. I start rambling off. So as we go, hit me with questions. Happy to share mediums. Happy to share protocols. If you guys have, take advantage of this because I charge a crazy amount of money for my class. There's a reason we charge, though. We charge because I'm confident that I'll make you successful. And I backed it up with my students and other people who've taken the class, and it's what it's all about. Anybody can learn this. It's just timing. If you want to speed up the process and move it along, you'll come take our class, get educated, and then have 6 months of follow-up to make sure that you can, pass over the speed bumps along the way. Because most people pack it up about halfway in. After the mounds of infections and beating yourself up, piling up dirty containers of failed cultures, you literally will park it. But not with me on your ass. I'll be on you. I'll be calling you. What's up? It's exciting when you finally go in and you see roots for the first time. You see an old plant that you fixed up and brought it back. I wasn't convinced until I put certain strains in. I had the banana OG. Everybody's heard my banana OG. But that motherfucker, man, was beat. That thing was done. And it came back like never before. This thing dumps almost 7% now. And I'm not trying to toot 7%. I hate numbers like that, but it does. People that are running this thing, it's like killer. I'm going to breed it with everything. To get those kind of numbers out of an old beat up plant, that was meristem. That thing came back through meristem culture. It took almost 2 years. Now it's fucking fire. I know this process works. And again, before I did that, I was kind of leery about TCS. Like, come on, man. You're going to get rid of virus. You're going to do all this crap. Give me a break. Now that I've done it long enough and witnessed strains that I know had problems and seen them grow and what they do, it's nuts, man. These plants it's night and day, man. Like I said, not only are they healthy, but the structure changes because of the hormones and the process. So it's like you can create super plants, man. It's nuts. Like I said, this is one of the funnest things I've ever done because we like being in control. We're not in control of the world right now, that's for sure, but I can be in control of these plants. I can literally tweak these hormones and do shit with these plants. And this isn't even genetic modification. This is what's nuts, is I want to go to that level now. Once you get to a certain point of comfort and your confidence builds, now it's like I want the pathology. I want to understand the DNA, the CRISPR technology. I want to take out certain sequences that are crazy elements and then put those into other strains. Obviously, I don't want to do anything that's going to compromise our health. I just want something that's going to produce yields and produce some terpenes that are measurably different. I've got something on my mind that's driving me nuts. Excuse me for 1 second. I really want to figure out testing. I feel like the testing right now that's going on in the industry is just BS from not hops and stuff. I think that Toomey's got their shit together, but certain other companies, there's no doubt. I feel like their testing sucks for potency, for everything. I just don't trust it. I think there's a better way to test, and it's going to have to come down to honesty because people getting paid off to bump the testing up and this and that. It's just bullshit. I want some actual real testing, and it's something that's greater technology. I feel like in the past it's been shit. So hopefully we can come up with some better testing mechanisms to actually determine what we're doing works. Only way to truly determine how something is successful is by measurement. This is what's a great thing about tissue cultures, because now having so many copies of a strain in culture all the time, I can run random tests from nutrients to hormones to products, you name it. I can just put the plant through different circumstances. It's pretty amazing. So the things that tissue culture has been able to offer for us is stop loss, increase production, and terps. I have no doubt, like I was talking about the banana, the terps are there. And when the terps come back after an old beat up strain, I don't know how old that strain was. I'm thinking 2000, maybe 2005. I'll have to ask Ed how old it was, but it was ancient. That was the original Oregon Kid banana OG, and it was beat, and it's back. So who's into tissue culture? Anybody into tissue culture, or you guys just into genetics? everybody just want to learn about fire genetics, or do you actually are people feeling the need to get educated on tissue culture? It's not for everybody. I can tell you, tissue culture is kind of nerdy in a way. You know, it's clean. Not going to get dirty out in the garden. It's not your average labor, which is to me what cannabis is now. It's a labor-intensive job, where tissue culture is more of a thinking now, where you're thinking about protocols and solutions, and that's really exciting because I feel like you can really alter the course of cannabis from this point. People who really have a passion for cannabis, for flower, for rosin, for food, for anything you want to grow. That's the beauty of tissue culture. It transcends cannabis. This actually can lead to other, ways of self-supporting yourself in a crazy world. You can keep fruits and vegetables for long periods efficiently. The beautiful thing about TC is the medium is made of sugar and water. One of the cheapest mediums on the market. Talking about tough times, go look at the prices of rock wool and soil and Oasis cubes. Sugar and water, still cheap. That's the primary base of what we're using. A little bit of salts, medium. I mean, it's a tiny 5.3 grams, I think it is 5.32 grams. It's a tiny amount for a liter. I can make 60 test tubes. Literally, I think it's roughly maybe 7 cents, maybe 10 cents a test tube. Still, by far, one of the cheapest methods of massive cultivation, not compared to anything. This is if you want to do 3 to 4, 5, 10,000 clones, sugar and water. And once we cut the plants and I put them into the test tube, the beautiful thing is there's no watering, no spraying, they're there. Once I put the plants into the tubes and they root, once they root, I can take them out, or I can leave them in there for up to a year. Awesome fucking thing. That's like one of the best things ever. All right, I'm already like 37 minutes into this, man. It's not even 10 o'clock. It's supposed to start at 10 o'clock. Is it cool that I started early? Again, anybody who's on, we're grateful for everybody who jumped on and subscribed. That's grateful. I'm going to try to be on here as much as possible, dropping knowledge. We've got lots of good stuff coming up with some students, and we're going to be sharing that information. Again, this is something, if you've ever had a passion to do TC, if you want to preserve some of your old strains you've been hanging on to forever, and you've been hanging on to them, and they're probably just scraggling, but don't want to get rid of them. I've been to so many people's grows, and they've got these plants, and they're their favorites. RS11, it's been around forever, but they won't let it go, and it's just done horribly. I don't know what it is about us. I'm the same way. Certain strains, and it's like I just can't let it go. But I got to tell you, man, once you take the chance, put them through culture, and I'd rather teach you than charge you. It's like 5 G's to put a plant through TC. It's really crazy, but when you see how much time it takes, it's sometimes up to 2 years to babysit one of these plants. I can teach you for what you would pay another company to do TC on your plant, and we'll teach you the process so you can do it yourself. Nobody's going to look out better for you than you. No one's going to give you a better deal than you. And it's better to keep your genetics in house if possible. Why not keep control of all your stuff? I don't care about people's genetics now. When people bring in stuff, I tell them to label it, change the labels, change the names. I don't care. I have so many strains. That's not my thing anymore. And by the way, we can add dye markers, DNA markers into the plants now. And you can identify the plants years down the road. If someone has a strain and they say it's theirs, you can literally put a marker into the plant, and it will stay into the tissue of the plant. You can test that plant down the road through multiple clones. You'll be able to test that plant and say, that's my strain, that's my marker. Because these DNA proteins are like little codes, So if we put one in a plant, it's very easily identifiable down the road. You can mark your strains and keep your genetics locked down. So the beautiful thing about doing TC is you can do all this stuff in a small area in your house, keep all your own genetics, or have a backup from your regular grow. So if shit happens or you lose it, or God forbid something happens, that's been one of the best things about TC. We had a fire at our lab and the whole upstairs caught on fire. And I'm in there under the hood working, and I have my headset in, so I couldn't hear, didn't smell anything, didn't smell smoke like a dummy. Fucking whole ceiling caved in next to me. I'm sitting there working in the hood, and the ceiling caved in on me. Pretty much water and smoke. At that point, I ran out, but it was awesome because the tissue culture wasn't damaged. The fire department went and got a covered up all my shit. And they wouldn't do that with your plants, I guarantee you. But because it was TC, They went and got tarps, covered all my shit. Nothing got damaged. I was able to survive basically a flood and a fire, and my shit still made it through. So whenever there's a problem, tissue culture, even if the lights go out because of the charcoal or because of the carbon, they won't die. They'll start to flower after a month or two, but they won't die. They can photosynthesize because of the sugar. They don't even need light. That's why such minimal light is required in TCs, because the sugar, carbon in the medium, allows them to photosynthesize. Hence, this is why I can cut them to nothing. No, the little tiny pieces with no leaf, they don't need leaf. They'll photosynthesize because of the medium. It's crazy. Okay. Looking at the comments. Bear with me for 1 second. Awesome. I'm glad we fueled your curiosity. That's what this is all about, man. We want a little spark. If I can spark one person with some motivation, some desire to get better, to do better, either in growing or their life, that's what we're going to do. That's what this channel is all about. We're literally want to share. I've got so many students now that have come through class and taken this class that we're creating a mastermind group, and we're going to be sharing protocols, information, ideas. I got to tell you, that's one of the best things about this. When we have a platform of people who are all, we're all on the same page. We have the same desire. So when we're all in a creation mode, and not everybody's in creating mode right now, man, I can tell you people are in hold mode, hunkering down, for the winter and going through Bidenomics, whatever, no politics, but everybody's hunkered down. And now it's like, this is a time when, again, study, make yourself better, improve. TC can do that for you. TC really, again, not trying to sell the class here. This is about when people decide they want to come, I want you to come to us. But there's no strings to everybody coming here tonight. I'm going to get you some genetics. I'm really excited about, during the holiday time, getting people some fire in their hands. All right. Yes. Learning, man. Right now, I got to tell you, whenever we're stressed out, when things are crazy, that book, 6 Habits of Growth, it says whenever you're stressed or uncertain or things are kind of crazy, literally to read, to educate yourself. If you can form a habit of literally every time things are crazy, things are stressful, to take 15 minutes and read something, it's usually the lack of understanding something. But anytime you can educate and give yourself knowledge during stressful times, just the few days I've been following some of the steps out of that 6 steps or 6 habits of growth, incredible. No social media. I do this to get posts and put stuff up. There's no social media in the mornings now. I literally will go to the gym 4 or 5 in the morning, work out for 45 minutes to an hour. Right when I'm done, I'm in my car. I'll either read for 15 minutes at that point. And when I walk out of the gym, sit in my car and I'll read. After I'm done reading, I'm usually home at that point. I'll write out my goals, have a snack in between, and then I meditate for 15 minutes on the stuff that I just set out in the goals. And it sounds crazy, but it really has helped me tremendously. It's helped me to be here, to be in front of you guys, to set some goals. This mastermind we're going to be setting up. If it's something that interests you, shoot us a DM. I'm really looking for people who've taken the class or are Black Sheep former members, people who've spent money with Black Sheep. And I only doing that because out of fairness, everybody who's in this mastermind group we're working on has got some skin in the game. So thank you, everybody who's showing up. I started early. Don't hate me for showing up early. Everybody who showed up early, give yourself a pat on the back. Most people in this industry are a day late, $1 short. No offense, but you're here, and I'm grateful. Everybody who went out and subscribed to our channel. This is going to be about sharing information. We're able to, through our other students and people that are continuing to create protocols in tissue culture and create better solutions, we want to share that. But it's only possible if you have, a lab or some understanding. I literally don't want to sell genetics anymore unless I can give them to people in a test tube. That way it kind of takes me out of the loop. By giving people plants in test tubes, there's no root aphids, there's no spider mites, there's no anything. It comes to you in a test tube, you're able to pull it out, harden it, and that way it's literally no 100% guarantee on my part that it has nothing when it's in the tube. Once it's out of the tube and exposed, just like anything, they're not super plants. They get exposed actually a little bit easier because they're bubble plants. They've been inside of a test tube. So you really want to keep them sterile after they come out of culture because they're susceptible. It's like a baby. They don't have an immune system. They've been protected inside of a container for so long. So I want to give them to people in the tube. It's preferable. Everybody who's jumping on, thank you so much for subscribing, following Meri Genetics. We're grateful. I've been on here for 45 minutes now. I got to jump on the gun, all excited. So if that's all right, it's going to be available for replay. Let me check out some of the comments here. Awesome, brother. Thank you, everybody who's been following, who's jumped on. I usually don't root the plants in, I start them in the test tube. I usually do the 50% sterile water, 50% peroxide. I try to make them pop out of the shell completely. At that point, I will put them into agar media, rooting media, and that takes anywhere from about 14 to 30 days to get them like a normal, I got some right here. Check it out. Oh. Here's some of them right here. You probably can't see them very good, but I've got about 15 seedlings in there. And this took about, I don't know, it's been in here about 2 weeks now. It's a little bit slower growing than normal. They don't grow quite as fast, which is a beautiful thing. We're always in a hurry for everything, but when you want to take a break or vacation, when things grow slow in agar, it's kind of a nice thing. It gives me more control. I can pull them out. As soon as I pull them out and plant them in soil, they'll take off. So if you want something to go fast, just take them out faster and put them in the regular medium and start hitting them with salt. They're going to go crazy. I prefer things to grow slow now. I take them out as I need them. So this has proven I can just go through hundreds of strains now, and I'm not under pressure to, oh shit, it's getting too big. It's because it's been in this medium with hormones. They tend to be very compact and short. So they actually are, when they come out, they don't get lanky. They don't get all tall and lanky. It's better. But it's more about the control of timing. I get to select them out as I need them. It works awesome. Super fucking awesome technique. And for saving old seeds, that sterile medium is because it's so fertile. If you can just get a tip to poke out, that's all it takes now. You can just get the tip, poke it in that medium. We get like 99% now success from some of these old seeds. I find that old seeds tend to have very hard shells that are fossilized. That's why the peroxide in the sterile water help to break that shell, help to soften it. But even more so, we'll add some gibberellic acid and a cytokine, kinetin of some sort, to help the plants. Hormones help pop them out of their shell. I find that the old seeds, that's usually the most difficult part about getting them to pop is the hard shell. It's like a freaking dinosaur shell that they fossilize. So the longer you can soak them, the better. Pop them out under a microscope if we have to. We'll scalp them and pop them out. Stick them in that medium. The medium is the key. This medium has allowed us to grow, not only tissue culture, but seedlings as well. And it's just a rooting media, TDZ, NAA, and endobutyric acid, TDZ, and NAA. 3 different types. There's 2 auxins and 1 cytokine. Normally, you don't put a cytokine on top of auxins. It's supposed to inhibit the rooting. It's good. So anyway, let's answer some questions. All right. Yeah, we got all kinds of free game in here, man. Free game, some free fire. I'm so grateful for everybody who subscribed. I'm not trying to get anything out of anybody. I'm simply trying to share information. If anybody in the future ever decides they want to get involved or they have questions about TC, they're going to come to us for answers. I'm going to have all kinds of cool stuff coming up here. We have TC kits. I've got a seed kit, all kinds of shit we're going to put on here for some merchandise that people want. But I'm more or less interested in sharing information that inspires you to grow, either in the garden or personally. Cool? Hit me with some more questions. Challenge makes us stronger. Just remember that. Challenge makes us stronger. So tissue culture is a challenge. It ain't easy. Anything easy is worth having. Learn this shit, because I tell you that the amount of time that I've been doing this, it's given me so much more opportunity. It's open doors. It's open doors for other things. And like I said, I feel like it's kind of transcended cannabis a little bit. It's got opportunities out of cannabis now. It's leaning more now into the medical. There's a lot of people who are wanting to do studying on other things besides THC, and they have to go somewhere to get plants. You think about it. People that are trying to actually study, get grants and the government to do study for cannabis. Where are they going to go get their samples from? if someone like you has a TC lab and you have everything that's been tested genetically, it's all stored, you have all of the database, that's a great opportunity to sell to a medical company or a medical team to do testing on individual strains. When I say transcending cannabis, there's lots of other opportunities that this could open up doors to. It's really incredible. All right, answer some questions here. I'll quit flapping. Thank you again for everybody who's on. Hit me with some questions. This is free knowledge time. If you haven't sent me your information on my DM or anywhere on here, send me some information so I can get you some gear out. Don't forget to do that. Tomorrow, that's what I'm going to be doing. I'm going to go over all of this stuff, get everybody some fire gear out for the holidays, okay? All right, let's see. It is, yes. Powdery mildew is a huge problem. Once you go through TC, the beautiful part about TC is the sterilization process. We run them through detergent for 15 minutes, and then we do an alcohol rinse for less than a minute, and then we do a bleach rinse. And that bleach, with a little bit of wetting agent, actually per minute, it gets into the plant. And a lot of people, there's a debate on PM. Is it internal? Is it systemic? I'm a believer. I think it is. I think it's both. I think it kind of intertwines in the plant's capillaries in the cell walls. So bleaching the plants for 15 minutes in that shaker, that orbital shaker that you see on my video, that's what it's doing. It's sterilizing the plant. And then by cutting the plant in such small pieces, you eliminate the bodily, the infection, most of the sick part, PM, whatever it may be. You get rid of that and regrow it in a sterile medium. Now, that medium acts like a sponge. The beautiful thing, it's not just food for the plant. It's actually like, because it's so potent, it's so fertile, anything inside of that plant, any kind of cellular bacteria, endophyte, once it comes in contact, it will grow into a plume. I've seen some crazy colors from red, pink, yellow, green, blue. That's really created the fascination for swabbing. I want to go into the pathology because I want to know what is coming out of the plant so I can create protocols to put into the medium to prevent it next time from happening. You really want this stuff to come out. It acts like a sponge. That's exactly what the medium is. The medium is a sponge, and it cleans the plants of internal infection. This is the beauty, because once it's clean, these things perform like you've never seen before. When you want a plant to grow like a seed growth rate, like it was from a seed, that's the beauty of TC. It really brings that to fruition. It's awesome. Hopefully you've gathered a little bit of info tonight. Thank you so much for watching, for subscribing. Great. Really appreciate it. Hopefully I'm not missing any questions here. Yes. Thank you, Top Notch Canna. Grateful, man. All we want to do is share information, man. If we can help one person, I'm super grateful. I want to help one person get better in cannabis, in TC, in life, then I'll be super grateful. I'm not going to go on for too much longer. I've been on here already for 53 minutes. Everybody who's here now, grateful, super appreciate it. Thank you, brother. Appreciate you guys watching, following. I'm going to have, like I said, I want to be dropping some new products coming up. Looking forward to talking about some of the new technology, some of the magnification technology, so we can do that. I don't know if you guys have watched the video, some of the videos I have, where you can actually see micrograph, where you inject a dye into the plant so you can see if there's a virus in the plant before you even waste your time doing a meristem. That technology is coming, so we can either, through pathology testing or microscopic testing, we'll be able to tell if a plant is worth salvaging. Instead of just cutting a bunch of meristems and hoping one comes through clean, you can actually do a lot of testing beforehand and save yourself a lot of time and effort. No, I have not done root TC regeneration. I have not done that. Some people have done that with leaf, with stems. There's a lot of different things you can do in TC. I've really kept myself focused on apical meristem. That's been my primary study. And then also what takes place inside of the test tube, the gases, etc. There's ethylene gas that's produced when you wound the plant and put it into an auxin. So there's things you can do to the medium to prevent that. So we've really spent our time primarily focused on the media in apical meristems, but there's lots of other things you can do. There's the embryo seeds, synthetic seeds. There's lots of different stuff you can do for TC. It's really cool. you know, I'm not going to talk about the bad about the TC kits that are out there, but don't waste your money. It's nuts. Those things are just a gimmick, man. I feel bad for people that invest their money in those things. It's just taking your money, man. They don't care about your success, because that thing they're selling, you can't be successful doing that. There's no microscope. You need a hood. I mean, you can do it without, but when it comes down to it, you really want to learn what the medium is all about. You know, you don't want to have it in a packet. That's just easy. You know, you want to understand why every strain reacts differently. When you can figure out the medium, it doesn't take that much. You don't have to be a rocket scientist to figure this out. You just have to have the desire. The medium, you know, you figure out the hormones, the cytokines, the auxins. That shit will make you freaking crazy smart, man. When you understand cytokines and auxins, now you can create your own rooting gel. You can make your own plant food. You know, you can make solutions to feed into your plants, your feeding solutions that can make them turn into like super plants. The possibilities are endless with culture. It's really crazy how much shit you can do to these plants. It's just a matter of, you know, do you want to? What do you want to do? Again, everybody who's showing up, grateful for your, I got shit. There's been so many subscribers tonight. I didn't have any idea you guys are going to clean me out of all my seeds, man. Awesome. That's super great. I'm happy that there's that many people out there that are interested truly in cannabis and some fire and educate themselves. So thank you for giving me the opportunity to be in front of you. I'm excited to share any upcoming information and events. Again, we're going to be having a mastermind group for people who've been to class, for former members. If this is something you're interested in, you feel like you can contribute some ideas and you want to better yourself, shoot me a link. We'll shoot you a questionnaire and see if you're a fit for us. We've only got 3 people in the group right now, and we're going to probably fill it up to about 12 and then cut it off. And in that group, you want to see some powerful shit. Submit your goals to a group of people, meet weekly, monthly, hold yourself accountable, get in the hot seat once a month, figure out your problems, how we can all come together as a group and help you. You'll be surprised what you can achieve. A lot of us now are doing stuff independently. Understand it's kind of a greasy, crazy world, but we need each other to move forward. We need to have each other in groups, support groups, mentorship, whatever it may be. I want to be a mentor to people. If I can teach people, you know, I'm 52, I'm getting older. I'm not going to be doing this forever, you know, I'm looking to retire pretty quick. So if I can teach people what I've learned in a short time, pass that information and watch them do better than me, that's awesome. That's the kind of shit that makes me excited and happy now. Awesome. You're hooked. One person. If one person's happy here tonight, my job's done. I want to make everybody happy, obviously, but come on, we know that's not possible. Wealth of information. Thank you very much. That's all I care about. I want people to gain some knowledge from this. Hit me with questions. You can ask media questions, sterilization questions, meristems, nodes, hardening. Throw the questions at me. That's what this is all about. This is a Q&A. Not only that, but you're going to get some fire gear. Everybody who subscribed and give me the opportunity to present my business, my platform to you, I'm grateful, truly. Like I said, I couldn't do this without support. You guys are awesome. I see the hearts going off. This is epic, man. Only sad thing is I'm going to be out of gear. You guys are cleaning me out of all my fire gear, but just do me this one favor. Everybody who's getting gear, I ask that if you find something crazy, give it back. Let's TC it. Let's breed it. Another beautiful thing about TC is I can keep these plants. I can keep males. I can flower a male plant in the test tube, and I can flower a female in the test tube. I can breed in the test tube. Fucking insane. I can actually breed test tube to test tube. And then I don't have to wait for the seeds to get mature. This is called green pod seed sowing. Normally you have to wait for the seed to develop, and that takes months. You know, it's normal 30, 60 days, but you can wait until the seeds just turn green. That little embryo, I can pull them off inside the tube. I only get a couple, but it's enough. And I can put them right back into agar and they'll grow, cutting off like 4, 5, 6 months of grow time. Green pod seed sowing. Look it up. It's incredible. So you can breed from test tube to test tube. I can keep all my males. I can keep, you know, a female and flower in the tube. It's nuts. If I want to do it bigger, I just use a bigger container. I can flower in a pickle jar if I want to and flower a plant inside of the container. I think that's coming more than likely. If you can avoid bugs and problems and all that shit and just flower the shit in a container and not have to deal with it, not have to water it, not have to spray it, it's fucking genius. You wouldn't want that. Sweet. If you want to order something, please give it back. You're going to find something. Some of the ones I'm sending out, I've got some of the old Sunday Driver wedding pie crosses. I don't have the Sunday Driver anymore. That strain was it was a cup winner, just a fucking killer, awesome, wicked strain. A lot of the ones I'm sending out are the Sunday Driver wedding pies, rainbow belts, banana OG, shit. Zoda Z Claire's, peanut butter rockets, marshmallow OG. Got some fire going out, guys. I'm excited to get it in your hands. Some of the packs I sell for up to $250, $100 average. I'm not saying, but I have value in my seeds, and I hope you guys understand that. I want you guys to grow some fire. Hopefully it'll change your life. Hopefully you'll get into TC or, something that improves your life or you improve the industry. That's what I'm looking for. I want to see people actually better themselves and do something for the industry. I think the industry has so much room to grow, no pun intended, but it's exiting. People are getting out. I saw so many people quit, getting out of this shit, and it's sometimes when the tough gets tough, the tough get going. I think that's it's a metaphor, but it's for real. Sometimes when things are tough, that's when you buckle down, man. Get smart, be prepared, because the market's going to come back. I tell you what, I'm going to set myself up so when the market comes back, I want to have every fucking strain locked in TC, all tested, pathology, just sitting there waiting for me. I can go on vacation and nothing's going to die. I can breed. I can, do testing. It's incredible. Like I said, I think going into the future, testing is going to be completely different, not only for hops and Fusarium, but for potency. I think it's been BS for a long time. The minimum cost for a small setup, dude, honestly, you can get a nice setup now. You're going to want to spend at least $2,500. That's the lowest. The biggest cost is going to be a hood, and you don't have to get a big hood. You can get something. They got these little small units, but you've got to have room to work. You've got to have room to put a microscope. You've got to have room to have your bead sterilizer and some of your medium. So, as much as you want to skimp, I saw people get good hoods for $1,000 to $1,500. A good autoclave is going to cost you around $1,200. Microscope, shit, you can get a good microscope for $500 to a G. Realistically, about $5,000. If you can come up with $5,000, $10,000 is going to get you everything, really nice stuff. But you're looking at about $5,000 minimum to get started. Our class, we're not cheap. We charge like $7,500 a student, and it's only for 2 days. It's not just the 2 days; it's 6 months of follow-up, and the guarantee that you'll succeed. It seems like it's not cheap, but either is my time, and either is my ability to make you successful. That's what I guarantee. When I'm taking your money, I guarantee you, if I charge you that much money, I'm the most expensive guy, I think, right now in TC. And it's not to be a dick. It's because I value my service. After the fact, I don't disappear. I want to make you successful. I want to make sure that you understand and become successful. I want you to become part of the mastermind group, and I want to learn from you. I want you to become smarter than me and pass knowledge to me. That's what's cool, is when the student surpasses the teacher. I want to do that. Black Betty, hell yeah, dude. Black Betties are wicked, man. That's like a Runts, Sunset, Skittles cross. Fucking nuts, man. I have some people who are running that in a legit facility right now, and holy crap, man. There's so many strains. This crazy part, it sucks that I'm so expensive because it limits people from having our genetics, but at the same time, it's like, it does create value in them, but man, I want to give them out. I have so many crazy heater strains that I want to get out, but I know everybody does now. Everybody breeds their own stuff. Everybody's doing their own thing. And hey, man, I support that. I support people doing their own genetic crosses and breeding, etc. I think that's one of the best things ever. I think COVID, out of all this shit, really created a lot of independent entrepreneurs, and hey, man, thumbs up to that. The only problem with that is I feel like if you got sick shit and you're breeding, then that's a problem. And I think, people don't know now. And if you don't know, understandable. But if you have an inkling and you got if you can see something, and don't be breeding that shit and spreading that around, man. I don't want to grow your bunk. I don't want to waste all that time. Hunting is a pain in the butt, man. That's why I love tissue culture in this process with seedlings. It really changed the game for me, because now I don't worry about them getting sick and the timing and the rushing, but I'm always hunting. No matter what, I'm hunting 50 to 100 new strains on a regular basis because I'm able to, because I'm not contaminating, because I'm not under pressure, because my seedlings are 15 feet tall on all lanky. So many benefits for TC. Get smart. Follow our page because I'm going to be giving out information on a regular now. Every time we have our mastermind group, once a week, once a month, I'm going to go on here and share the information. We're not going to keep our secrets. I want to be a gatekeeper. I want to give you guys information and help you succeed. That's what this is all about. Giving you fire gear hopefully helps you make some money so you can save, come to class. Grow out some fire gear, sell some cuts, whatever you got to do, save some money, buy some equipment, learn TC, change your fucking life. It's that simple. It really is. When I wanted to learn TC, man, it was like, it wasn't that long ago. And looking back now, it's crazy because now I want to expand way farther. TC is awesome. It's great. But it takes you a good amount of time to get comfortable. Once you're comfortable, though, now you're talking pathology and DNA and really getting into some exciting times that I think we can create something special. Not only potency, but specific flavors. And I don't know, we can just really boil it down to perfection. And we can do this without football size grows. Don't get me wrong. If you got a football size grow, I'm all for that. Cool. You need a lab. Hit me up. But the small guys, the craft farmers, that's what I want to support. I want to support the guys who grow the fucking fire, man, who grow that weed. This is why I want to help because I love weed so much. And as I get older, I'm not going to grow. But I'm counting all you dudes, all you fools who grow this fucking weed. I'm going to be hitting you for some fire. So take this fucking shit I'm giving out, grow some fire, get some knowledge, hook a brother up. Again, challenging times make us stronger. Okay? So I appreciate you guys giving me a challenge and giving me an opportunity to get in front of you. We've had a good amount of people come on here tonight. I can't tell you how many people have signed and subscribed tonight. We gave away some genetics. Hey, whatever, you have to pay for advertising and marketing now. I have no problem doing that. But in the meantime, I was like, I honestly didn't know, how many people were going to be interested. And this is incredible. All the support, all the people coming on. I can't wait to get you the gear. That's my mission tomorrow. Don't just shoot me your information. I'll get you some gear out. Sorry, I've been just rambling, man. I'm just excited as hell to be able to share. Again, if it's one person I can inspire, one person I can motivate, that's what I'm here for, man. Check out the book, 6 Habits of Growth. Brendan Bouchard. Dude, fucking epic. And I don't recommend books. I'm not trying to be a motivational speaker, but I'm telling you, it's a quick book. I've already read it like 6 times, man. And the more I read it, the more shit I gain from this. But it's my morning ritual has changed my life. It's allowed me to get in front of you guys, have the courage to sit here and talk, be faithful in my conviction, in my encouragement of you, and knowing that we can be successful. We can accomplish more together. Not trying to sound cheesy, man. I'm truly inspired by everybody who's signed up tonight. Contact me on there should be something on here, hopefully on YouTube. But if not, just hit me on Meri Genetics on Instagram. Shoot me DM. Or you can go to our website, merigenetics.com. My phone number's on there. I'll put my phone number on here. Anybody who wants to contact me. I'll make it easy for you guys to get a hold of me. And again, you can call and ask questions anytime. I'm super grateful for everybody who showed up. I've been on here for an hour and 8 minutes. Got on here early. Super grateful everybody who showed up. This has been way more than I expected, man. I'm so honored and intrigued by everybody. I can't wait to share more information. I'm going to spend all day tomorrow getting gear ready to go. So don't forget to give me your info once again. I know I'm double talking here, but I want to make sure that I don't forget. Your gear is my priority. Everybody who took the time to go and subscribe to my channel, fucking A, man. I'm here. I want to help now. Now you got my attention. So stay attentive to our platform now. Now that we got this set up, you guys can come watch, gain some valuable info. Hopefully we can provide some valuable stuff. I'm not going to ever sell you shit on here. This is about learning, educating, and in the future, if you do have questions, if you do decide to come to class, I want to be the one you go to, simply by trust, not because I'm, here selling you, pitching you. I love cannabis. I've been doing this. Shit, I'm 52 now. I've been doing it since 12. Smoking weed when I was 12. I know it's fucking horrible. Whatever. I'm a long-time cannabis enthusiast. I've literally been employed by cannabis my entire life. It's benefited my life tremendously. I want to give that back. Everything I've gone through, everything I've learned, ups and downs, crazy shit, anybody that I've had beef with in the past, if I didn't get your order right, if I screwed your gear up, if you got something for me that was sick, I'll make it right. I'm here to make amends, to be a better person, to be an honest, upfront. Like I said, I value everybody who's given me their time, who trusts me to sit here and listen to my spew. Hopefully I haven't rambled on too much, but I'm super grateful. I truly love what I do. I love that I'm able to share it. Like I said, I think challenging times create strong men. We're going through that. Don't give up. Get strong. Read some books. 6 Habits of Growth. Brendan Bouchard, game changer. Take these genetics I'm going to give you, grow some heat. If you find winners, I want to cut back. Mainly, make some money. Do something, you know. I'm not encouraging anything illegal here, but make some money. Buy some equipment. You know, for yourself, a TC lab is one of the cheapest investments in cannabis. And I assure you, if you put all your strains, we can save the cannabis industry, because all these genetics that are out there, there's a lot of ones that are shit. But if you can figure out how to pop those old seeds and then keep them preserved in TC without getting them sick, you're doing the industry a favor. And when you can provide a service to other people, when you can help other people, man, you want to talk about fulfillment. Money's great, but it only gets you so many endorphins and so happy. Listen to Dan Blazarian. After a while, you know, you have to find other means of fulfillment. And I'm finding mine in helping others achieve success. I want to know what you want to achieve, what goals you have. How can I help you get them? Through strains, through a little bit of personal motivation. Watch our channel, stay tuned. If you feel like joining the mastermind group, shoot us a link, a DM on my phone, the Meri Genetics. You can find a way to get a hold of me. We're going to have a small, tight-knit group, but I tell you what, it's going to be an honor and a privilege to be a part of that group, because there's some hungry dudes that are on this group right now, including myself, and we're going to bring ambition, and we're going to bring goals, and we're going to bring some stuff that'll hopefully motivate you and hold you accountable. Really important nowadays to actually have someone give a shit about your goals, not be so self-absorbed about us ourselves, but care about the group. And I really think we can inspire other people to do some good stuff. Again, super stoked. Thank you guys. Everybody, I'm trying to acknowledge you. Joseph Sauce, Captain Kindbud, everybody, thank you. South Jersey Genetics, Jose, John, Chris, everybody. You guys, grateful, man. I'm so grateful. I took the time. You took the time to listen, subscribe. I've been going for an hour and 12 minutes. I got cotton mouth. I need some water. Stay tuned. Don't forget to hit me with your info. Oh, dude, I'm going to give you some fire. I can't wait. I'm excited, man. Like I said, if you guys have a preference too, let me know when you send me your info. If you prefer regs or fems, I've got both. I'm going to work on that tomorrow. Again, grateful. I had this showing tonight that showed up. I don't want to get off, man. I want to keep giving you guys knowledge, but I don't want to, you know, drag you on. Probably want to go have a dab or got to deal with your old lady or something. Get up in the morning, go to the gym, read a book for 15 minutes, write your goals down, meditate. I tell you what, your shit will change. Thanks again, guys. Hit me with your info. It's been awesome. Thank you for giving me the opportunity to speak on this platform. Stay touched. See ya.